I see 9:30, so I'm not going to wait for the gong, pretend that it happened. Welcome to the symposium. This is an introductory talk about predicting evolutionary responses to a changing world. And as we heard yesterday from Brian and Marius' symposium, there's quite a bit of interest and variation in the way people think about predicting evolution. And it actually came into play at the nexus of this symposium, where initially two different proposals, one about forward-looking ENR experiments, evolve and resequence experiments, and their ability to predict, and one about using genotype-phenotype linkages through genomic prediction and genomic offset. And we merged them together around this common theme of trying to predict responses to a changing world. So in this talk, I will go through some sort of basic background about contingency and determinism, about predictability, about the questions that are being used, about the approaches that are being used, and then switch into an advertiser for some of the awesome symposium talks that are coming up later, and I'll give some previews through some vignettes of things that are coming later in the day. So I think we can't talk about contingency and evolution without talking about Gould. Look at that magazine cover, amazing stuff, amazing writing. This is an era gone by, but it is true that even dating back to Darwin's own personal correspondence, there was discussion about laws in evolution and whether they exist. But Gould's quote definitely was, as he was wont to do, the most eloquent, replay the tape a million times, doubt you get anything like humans. And that is contingency in its truest form, not even 1 in a million, a real doubt. So our evolutionary trajectory is wholly contingent. If they are not, when and why might deterministic patterns emerge? And this has been a question that's been studied really well through studies of repeated and convergent or parallel evolution. First morphologically, but then confirming that this is truly independent origins from comparative methods. And I like this quote from Blount et al. In 2018 that says, "The power of natural selection to find the limited set of high-fitness solutions to the challenges imposed by environments could, in principle, make those outcomes deterministic." And I think it's not only selection that could produce patterns of repeatability or determinism, but I think oftentimes we are implicating selection when we see those repeated patterns. So why do we want to predict? What is the goal of prediction? And I think Brian summed this up yesterday very well in his symposium capping talk about predictability as the ultimate demonstration of understanding mechanisms. So if we truly understand at a complete level, and if you abstract it out and we have truly complete data, this would be the crescendo. It would be the end game. We would understand because we understand it all. And I think that, in my mind, is absolutely the long-term goal. And there have been some really cool papers that have discussed this both philosophically and empirically. One was Marius and et al., including Brian, talking about this through the mechanism of speciation and the speciation machine, which I thought is a really clear way of thinking about the ways in which determinism could emerge and the ways in which you could study prediction. Our focus here in this symposium is certainly related, but it's a little more narrow. I think most folks that are presenting today are thinking about predictability as motivated by a need to provide information in the near term, perhaps even with a management-focused lens. What can we do with prediction today? What can we predict over the next several generations? What kind of information can we glean from our current day understanding and current day knowledge? And the reason why we want to do that is large part because of predicted biodiversity loss. So this figure here shows projected biodiversity loss by taxa over the coming century or this current century based on climate projections. And they see that, or they conclude that 1 in 6 species are going to go extinct under current climate policies. But it's pretty clear in this paper that they're not accounting for any evolutionary processes. And they're upfront about that, in part, I think, because it's hard to predict how much, and also that data is not available. But there are ways. I think there are folks in the symposium that will be talking about ways in which rapid evolution could change this story in some way. So in which rapid evolution could change R naughts from below 1 to above 1, and whereby you get evolutionary rescue. I think the other part about predicting that we may hear some about today is predicting evolution ways to slow it down in cases where we have unwanted evolution, like in resistance. So prediction in the context of rapid environmental change, that's what this symposium is about. And the general milieu, I think, is anthropogenic impacts are shaping environments and selective landscapes. There are considerable questions about the ability of populations to adapt in response to these increased rates of disturbance, but also not accounting for rapid genetic change when considering management options, when projecting biodiversity losses, when thinking about populations going forward, is leaving knowledge and potentially tools unused. So what can we do today? What are the tools we can use? How much can we predict over the near term in response to rapid environmental change? So I think if we're focused on this question, on the immediate utility, perhaps predictions made through correlations are the best immediate way forward for many systems, perhaps. And we'll hear lots of different perspectives about that today. So I've gone over 5 minutes, and I haven't really defined predictability, and I did that because it's hard to. There are lots of different operational definitions and working definitions of predictability in evolution. One, I think the most common is patterns of non-randomness in phenotypic and genomic change, largely used synonymously with repeatability. And I think that's a common one, and I understand why that is the most common one, and I've used it that way myself. Other ways to think about predictability are identifying factors that will influence evolutionary outcomes. That could be quantitative genetic factors, that could be specific agents of selection, and it may often be in the context of responses to environmental change. Another common one that I think is separate but related is prediction of phenotypic breeding values based on genotype-phenotype associations that can be extended to predict adaptation or more often maladaptation, and that is sometimes called genomic prediction. And the last one is an ability to forecast the pace, direction, or genomic architecture of evolutionary change, or even, when people are including demography, the probability of persistence for populations inhabiting changing environments. So I think those are some of the working definitions that people are using now. And before I jump to vignettes about talks that are coming up, I want to give my own little piece of my own story from work that was done in my lab. So in my lab, we oftentimes do evolve and resequence experiments where we track evolution as it occurs. And we most often do this with Drosophila melanogaster. And so the general setup looks something like this. We start with genetically diverse populations founded into replicate cages, and we track evolution in response to environmental change. That by track, I mean we do pooled sequencing across time to track changes in allele frequencies. I also mean the part that takes way more work is repeated common garden rearing and measurements of phenotypes in those common gardens. So measuring genetic change in phenotype over time. And we do these experiments in lots of different flavors. Much more recently, we've focused on manipulating aspects of the genetic composition of populations to see how that influences evolutionary outcomes. But for here, I want to focus on a project where we were explicitly interested in predictability. Predictability using ecological framework. So one that we were using is the life history theory, which is used often in evolutionary ecology. And we were talking about this, or using this juxtaposition between fast and slow life histories to predict evolution. So fast meaning you reproduce, there's a lot of energy towards reproduction. It comes at a cost often and stress tolerance. Slow meaning low reproduction but high stress tolerance. In this case of these seasonal experiments in Drosophila, we were predicting that there would be evolution towards faster life histories in the summer and towards slower life histories or enhanced stress tolerance in the fall. The other predictive framework that we were using from an ecological perspective is one that's somewhat less used in evolutionary ecology, and that's the ecological stoichiometry framework. So ecological stoichiometry considers the fluxes of elements between organisms and environments as a way of understanding oftentimes populations or ecosystem functions. And there's a great set of tools for ecological stoichiometry. I think it's a really interesting way to try to solve the phenomics challenge of studying evolution. And there are some great hypotheses as well. So one of them is the growth rate hypothesis, which is put forward by Elser some years ago, that increases in growth rate should be associated with increases in relative phosphorus content. And so we were predicting that we were going to see increases in phosphorus content in summer, followed by decreases in fall. But some of these stoichiometric techniques also allow you to measure changes in things like alkali metal content, which is really important for insects, ectotherms, maintaining ion homeostasis in response to cold. So cold's a major stressor for ectotherms, especially ones that originate from sub-Saharan Africa like melanogaster. So we were also wanting to test whether there was predictive change in the direction of evolution of alkali metal content. First, life history theory, we see rapid evolution, and it looks like this. So each of these little gray lines is an independent population. The black line is the mean of the populations. We see evolution towards increased rate of larval development, towards a faster life history, and then a really sharp decrease, so up and then down, just like we predicted. Stress tolerance shows the opposite pattern, down and then up, so an evolution towards reduced stress tolerance and then increased stress tolerance. Stoichiometric traits, also predictable, slightly different scale, but also predictable. So this is carbon to phosphorus ratio, which is the way they report it in the stoichiometric literature. So going down is more phosphorus, so initially evolution towards more phosphorus and then evolution towards less relative phosphorus. That's the fast versus slow transition. Alkali metal content, this is potassium. So initially a big increase in potassium, then a big decrease. We had slightly less strong predictions about this, but this is what you expect under cases of adaptive plasticity. You would see a decrease in alkali metal content outside of cells. Also wanted to ask questions about the predictability of the genomic architecture. And prediction here, we mean trying to identify a priori which genes or even SNPs are going to be involved in adaptation over seasonal time. And so what we did was we used prior experiments that were similar. We used genotype-phenotype mapping, of which there's no shortage in melanogaster, and knowledge of the molecular pathways associated with ion transport. So this is a Manhattan plot, one of many, but just one I'll use, that it was over the course of the whole study. This is changes in allele frequencies from August to November. And everything that's high in this Manhattan plot is representing parallel changes across populations. So if we color these dots by whether they're located in genes associated with prior genes implicated in seasonal evolution, genes associated with variation in fecundity, with starvation tolerance, or genes that are in known pathways for ion transport, we get lots of pretty colors on this Manhattan plot. And this is just gene-level predictability. But the reality is this is not enrichment. So if you do a hypergeometric test, if you test for enrichment here, there is no emergent predictability, even though this looks very colorful. It's more obvious at the SNP level, where there really is no predictability at all. And so we see lots of predictability from ecological frameworks. They're pretty useful at predicting the direction of phenotypic change, even trade-offs, even complex physiological trade-offs. We see the genomic architecture is largely unpredictable, even using prior similar evolution experiments, genotype-phenotype mapping of traits, that we see strong selection, we see strong adaptation, and we don't see a predictability in the architecture. What we're doing now is asking whether that unpredictability at the genomic level is a function of the quality of our predictors, like can we map better and then get predictability, or is it just true redundancy? So we're working on finding that out. All right, now I'm switching to Hype Man. So we have a bunch of great talks today. I want to highlight a few of them. At 4:45, we have Alex Rego, who's going to talk about an experiment in which they used variation both in the strength of selection and in the genetic composition of populations to test how both of those things interact to shape predictability and repeatability in evolutionary trajectories using Callicebrucus maculatus. That'll be great. One of the co-organizers of the symposium, Moi Exposito-Alonso, will talk at 4:15 today describing this green net experiment, in which is a massive global evolution experiment run in parallel, starting with lots of diversity in Arabidopsis, and determining how predictable the evolutionary responses are based on local climatic conditions. At 12:15, we'll have Emily Harmon presenting an experiment on plastic, the way in which plasticity predicts population persistence across new environments. So using an experiment with rotifers, looking at the relationship between the magnitude of plasticity and ultimately how that influences population persistence. And the last one I'm going to plug while I'm up here is from Daniel Anstett, in which he did both a spatial and temporal analysis of mimulus in California in response to drought over an 8-year period. Really focused here on evolutionary rescue. They find that mean population growth rate is correlated with the strength of selection, and that genetic variation at climate-associated loci is predictive of population growth rates. So that's sort of spanning landscape genetics all the way to rescue. The last thing we'll do in this symposium today is leave a section that's open, a time to talk about, to have hopefully a discussion about predictability, and we want to leave it open. So we have questions, some of them are up here, but we're hoping that other questions emerge during this symposium. So if you have something, a question you really want to talk about it as a group, you want to pursue it, please show up, and we'll use 15 minutes and hopefully long after that to try to figure out some answers. And with that, I will thank my co-organizers, Moi, Katie, and Stephen Keller, and I'll thank my funding source and also plug that I am recruiting people. So I'm going to play my little video here of people at our experimental site going in, braving the hordes of Drosophila that are inside of there. Thank you. All right, next up we have Diana Rennison with genomic signatures of climate adaptation in 3 spines. Take it back.
All right, thanks so much to the organizers for giving me the chance to speak to you all today. I don't think to this crowd I really need to emphasize the fact that temperatures are increasing and across many different habitat types. The world is getting warmer, and a lot of attention has been kind of focused on marine environments, but there's emerging evidence that not surprisingly, temperatures are also increasing in freshwater habitats. So many lakes and rivers are experiencing large increases in temperature. And there's a lot of evidence to suggest that there's a very high extinction potential for freshwater organisms, particularly vertebrates. And in order for these species to persist, they're going to require adaptation or dispersal. So we really need an evolutionary framework in place to better understand how we can predict organismal responses to climatic change in freshwater systems. And really the ultimate goal is this genomic predictive framework that was just beautifully outlined in the previous talk. And so there's a lot of different types of data that we need to integrate to really have a robust framework for making predictions. And I don't have time to go over all of these today, so I'm going to focus in on one area, looking at genetic architecture and how we can kind of incorporate that into predictive models. And the system that I'm going to leverage to kind of look at this is the three-spine stickleback. And it's a really beautiful system for asking these questions for a couple different reasons. So it's repeatedly colonized newly formed freshwater environments. It has a very broad range throughout the northern hemisphere, where it exhibits a diverse array of phenotypic traits. And we have really great genomic resources and high-quality reference genome. And across this really broad range, there's a highly variable climate with multiple independent climate clines. So the aim here we really want to address is the predictability of climatic adaptation by linking environmental factors to the underlying genetic architecture. And to accomplish that, we want to look at different independently founded populations to ask, is there repeatability? And in stickleback, they're kind of a classic case for morphological parallelism. And so we wanted to see, does that same pattern extend to environmental architecture? So our approach, just briefly, is to sample stickleback, not surprisingly, survey them across this broad climatic gradient, to aggregate climatic variables using a variety of online databases, including WorldClim and TerraClim, and a variety of different variables. We then input those into a PCA to reduce the dimensionality of these data and prepare whole genome data. And then we kind of use a standard bioinformatic platform to filter reads and call genotypes. And then we perform a Baypass analysis, which is an environmental GWAS approach to identify candidates underlying this association. So the data for an individual SNP kind of looks like this, where you have multiple independent populations that you've surveyed for a given temperature variable, and then you find for an association, all of the populations at, say, a high temperature exhibit a certain genotype, all the populations at a lower temperature have an opposite genotype. And when you kind of summarize this across the genome, you end up with a Manhattan plot where you have certain regions with a strong association exemplified by this Bayes factor. So once you have your candidates, then you do take kind of the standard approach to look at what genes are found within these regions and do kind of the downstream analyses. So our data look like this. We had populations going all the way up to Haida Gwaii in northern British Columbia, down to Baja, Mexico, along the west coast here. And we had 20, I'm just going to focus in on 2 regions of our survey because there's actually evidence that these are independent comparisons. So we had 29 sampling locations in the Haida Gwaii archipelago up here, and 87 sampling sites in California down here. And when we undertook a structure analysis, there was strong evidence that there was independent clustering of the sites within these 2 regions. So we have these distinct population clusters. They have an independent evolutionary history, and they also represent kind of extremes of this climatic distribution in terms of both temperature and geographic separation. So now I'm just going to give kind of an overview of the climate within these 2 different clusters. So here we have a map of Haida Gwaii, and this is an island archipelago. And it has kind of this classic western side and eastern side that differ in kind of key climatic variables, including temperature. So we have the western side being colder and wetter, and the eastern side being drier and warmer. When we conduct a PCA analysis incorporating all of these different climatic variables, we see strong separation of the western and eastern sides along the first axis. So here we have a map of our sampling sites, and you can see that the western sites all cluster over here, and the eastern sites over here in terms of their climatic variables. So this is not genomic data, this is climate data. And the first axis is explaining more than 57% of the variance. And we have the second axis here explaining 18%. So we're capturing a large degree of the environmental variance just in these 2 dimensions. And that's important because that's what we're really going to map in our genome-wide association. Here we have California, and again, there's a gradient, but instead of being west to east, it's really north to south. So in the north, we have a much colder and wetter climate compared to the south. And you can see there's also a west to east gradient, but the stickleback in California are largely found very coastally, with the exception of this one inland population, which is an introduced population. So what's really key here is this north to south gradient. And you can see when we input this into a PCA analysis, we have segregation along the first axis with regard to latitude. So we have our northern populations over here and our southern populations generally over here. And again, these two dimensions are explaining a large fraction of the variation. All right, so we conducted a Baypass analysis using this PCA 1 and 2. And then the resulting Bayes factors were averaged across 10,000 base pair windows for all of the SNPs. And to be considered an outlier in our analysis, we considered windows that had a mean Bayes factor and a mean correlation coefficient above the 95th percentile. And this is just the average Bayes factor plotted for the Haida-Gwaii data across all of the chromosomes in the stickleback genome. And you can see the windows indicated in black are those that were significant outliers. And so the first thing that you'll just notice is there's not this one region of the genome that explains all of the variants. It's very polygenic. So all of these different chromosomes are contributing heavily to the signature of association in PCA 1 and 2. So now the next few slides, I'm going to show you kind of a more higher level summary of the signatures in the genomes. So here what I'm showing is on the x-axis the different chromosomes in the stickleback genome, and on the y-axis the number of significant candidate windows on each of those chromosomes. And so the first thing I want you to notice is just that all of the chromosomes had significant windows. And the ones that are indicated with the asterisks, these are windows that were significantly enriched for outlier windows. And so you can see there's a handful of these chromosomes that are disproportionately important to the genetic underpinning of this environmental association. And that was determined through permutation testing. And these data here are for the Haida-Gwaii archipelago, but we did all of the same analyses for California. So here we have the same summary of the chromosomes on the x-axis and the number of candidate windows on the y-axis. And again, we found enrichment on a handful of chromosomes. So the next question was, are these the same windows between these two different comparisons? So we overlaid the candidates, and so we looked at which ones were shared between California and Haida-Gwaii. And we have all of our chromosomes and then the number of shared significant candidate windows on the y-axis. And so you can see some of the chromosomes that had unique candidates, had candidates are not shared between the comparisons. So chromosomes 1 and 2, although they do have a genetic underpinning for the environmental association, they were not shared between California and Haida-Gwaii. However, there's other chromosomes that did have shared candidates, and in total there were 60 shared windows between the California and Haida-Gwaii comparisons, and that was more than we expected by chance through permutation testing. These candidate windows contained 116 gene accessions, and 29 of those were well-described and named genes. So now I'm going to take a moment to tell you kind of an interesting potential adaptive story that emerged from these candidate genes. So there was a number of exciting candidates, but one was this gene here, which I will not attempt to say, that is a eukaryotic translation initiation factor. And when we found this in one of these shared windows, we were pretty interested because this gene has been shown to be important in salmon. So in 4 different salmonid taxa, there's evidence of differential expression of this gene when the fish have been exposed to thermal stress. So that's pretty interesting. And then in ice fishes, this gene is actually contained within a region of the genome that shows the strongest signature of positive selection. So across this really broad taxonomic scale, we have evidence of an association between environmental factors or temperature and this candidate, which is shared between the California and the Haida-Gwaii populations of stickleback. So that's pretty interesting and something that we really want to validate potentially through experiments and our own differential expression analyses. A number of other interesting genes kind of popped out, including others known in thermal adaptation in trout, some known for photoperiod associations, and several immune and wound genes, which will be very interesting to kind of follow up on. And of course, you're kind of required to do some sort of Go-term analysis if you've done a genomic scan. And so we did that, and we found 3 different Go-terms that were enriched. I have no idea what any of these do. If you have seen these before in your analyses, please come talk to me. But I would just thought I would throw them up for you. Okay. One of the great things about stickleback is we just have so much pre-existing genetic data. So Peichel and Marquez in 2017 summarized all of the quantitative trait locus data for stickleback for all of the morphological traits, life history traits, so on and so forth. And what I've done here is kind of lined up our candidates for California, for Haida Gwaii, and those that are shared across the chromosomes. And these chromosomes here have been shown to be enriched for a variety of ecologically important traits. So those underlying feeding and body shape and defense and swimming. And you can see they're also enriched for our environmental associated candidates. And what's really interesting about this is that this genetic architecture of the environmental association and the genetic architecture of these other key traits could actually be either facilitative or constraining in terms of the adaptation to climate. So it will be really interesting to look at kind of the associations and how they play out once we start digging into them. The other thing to really take away here is that the large fraction of the architecture in either of these comparisons is unique. And so we do have some predictability of these shared candidates. However, the large fraction of the candidates are different between California and Haida Gwaii. And so we might be able to make some predictions that extend across the wide range of the stickleback, but we might have to take really more smaller scale approaches when trying to make clear predictions. Okay, so my take home is basically that the genetic basis of environmental adaptation is highly polygenic, that there's enrichment in certain regions of the genome, and those overlap with regions we already know are ecologically important in stickleback. Most of this architecture is unique, but there is a significant portion that is evolving in parallel, and that is quite interesting. And we have these exciting potential candidates that we plan to explore more. So we still have a lot of work to be done. We really want to integrate this into a genomic forecasting framework to work on our functional validation, do more expression studies, and incorporate our estimates of genetic diversity to look at this predictability and this forecasting. And first and foremost, I have to acknowledge Caitlin Wise. So this work is really hers. Anything that I've gotten wrong, that's on me, not on Caitlin, just to justify that. And many people have contributed samples to this broad geographic sampling, so I'd like to acknowledge them, as well as our funding and you for your attention today. Thank you.
So the next talk is a recorded one by Brendon Lynn, and it's Prospects and Limitations of Predicting Maladaptation to Future Environments with Genomic Data.
Increasingly novel climates to many ecosystems across the globe. This is data from a paper from 2017 that shows climate novelty across North America. But what I mean by climate novelty is the multivariate climate with no analog in the location's previous history. So of course this is going to create biological challenges, particularly for species that are locally adapted to climate. I study forest trees, and so as an example, without any climate-aware intervention, forest productivity could severely decline, seeing decreases upwards of 50% in California alone. So this type of information that is used to make this forecast come from reciprocal transplant designs, which are often resource and time intensive, particularly for long-lived organisms like forest trees. And so over the past decade, people have turned to genomic data to model relationships with climate of natural populations, and then to predict their vulnerability from climate data, which can come in hand much sooner than these transplant experiments and inferences from them. These genomic methods that I mentioned are generally referred to as genomic offset methods. And ultimately, they ask the question, what is the genetic change necessary for a population to maintain climate adaptation? These models are created by sampling genetic and environmental data multivariate environmental data from the landscape, and there's all sorts of different kinds of offset methods, but each of them create a model that relate genetics with climate. And then for a given population, we know its current genetic composition and its current environment. And given some forecast of climate change, we can use that model to estimate the necessary genetic composition of that population in the future. And the change in that composition is what is generally referred to as genomic offset. So a large genomic offset means that population will need to change quite a bit to maintain that relationship, which is increasingly unlikely as that genomic offset increases, meaning that that population could be more vulnerable. And of course the opposite is true as well. If there's little genetic change needed, then that population isn't inferred to have much climate change vulnerability. So over the last decade, these methods have increased in use and in numbers. But despite their increase in popularity, there are only a handful of cases where folks have tried to verify the predictions from these methods. This is a paper that we published this year that came out in Global Change Biology. And I can't get into it too much, but what we showed with empirical data from 3 conifer taxa is that these gradient forest models do well to predict performance in a common garden, but there are caveats. And I don't have time to enumerate those caveats, but what those caveats add up to is the conclusion that the domain of applicability for these genomic offsets is largely unknown, meaning we do not know the circumstances that they'll do well or the circumstances that they'll do poorly. And so the domain of applicability refers to the circumstances under which a method is acceptably accurate. And for genomic offsets, that domain encompasses the evolutionary history of the targeted populations, as well as the experimental design that is used to train and validate the model itself. One of the reasons that these validation attempts are so few is because the data necessary to validate these models is also rare. But fortunately with simulation data, we can kind of sidestep the weight and the resources and generate data to test these methods in silico. And so with these simulations, we want to create circumstances that represent biological realism, contrasting cases, and adversarial scenarios. So biological realism refers to the simulated data reflecting patterns in empirical data sets like environmental climes. Here I'm showing just sea surface temperature. These simulations should present contrasting cases like varying population connectivity but maintaining some average population differentiation, and present adversarial scenarios where data sets capture extreme characteristics of empirical data that could challenge these methods, such as predicting to novel climate change scenarios. So with this in mind, we created simulation data sets that tried to capture these 3 goals. So over the next few slides, I'm going to be explaining the scenarios that we simulated. The first one is the arrangement of environmental data on the landscape. So we simulated 100 populations with at least 1 or 2 environments to which the population is adapted. The first landscape is a simple Klein scenario where we have a temperature-like environmental variable that has a north to south Klein, and then a second environmental variable that has a Klein from east to west. We have a mountain scenario where that second environmental Klein represents elevation. We still have that temperature gradient though. And a third scenario which is representative of estuary Kleins. And what we were trying to get at is simulating an environment where there's a marine environment that populations have connectivity, but inlets that have Kleins in salinity. So with these 2 landscapes on the outside, we have geographic distance corresponds to environmental distance. We might expect these scenarios to perform well with genomic offset methods. But here in the center, we have populations on either end of the landscape that have the same multivariate environment. But of course, even with isolation by distance, we expect the genetic composition to be changed. And so will that present a challenge to methods? So the next few levels of the simulation, I'm going to go by rather quickly because these levels didn't show to have much effect on performance. But we did, like I said, we had 1 trait that was the environment that populations adapted to, or in some cases 2 traits were adapted to. And in some scenarios, we had equal selection on those two traits. In other scenarios, we had unequal selection strength. And then in other scenarios, we allowed for pleiotropy in the same type of situations. We also varied population size and migration connectivity. In several scenarios, we varied the level of polygenicity, signifying the number of loci that underlied the adaptation to those environments, ranging from a few loci to many thousands. And we tested 5 implementations of 4 genomic offset methods that have become popular in the literature. And what I want you to take from this slide is that 4 of these can take multivariate climate data, while 1 cannot. And 2 of these can allow for population genetic structure correction, while the others don't. For the genetic data that I provided, in the paper I provided the whole data set, all loci. Since it's simulated data, we know the adaptive loci, so I could provide that as well, or the neutral loci. But today I'll just be talking about all or the adaptive loci. And then for the climate data that was provided to the models, we used the adaptive environments only. To quantify the performance of these models, we created a reciprocal transplant design across each population on the landscape. And we projected offset each model to each of those 100 populations' home environments. And then in each in silico common garden, we measured performance as the Kendall's tau relationship between fitness that was known and the offset that was predicted. And so for a well-performing model should have a strong negative relationship between fitness and offset, so that high offset indicates low fitness and low offset indicates high fitness. So across all these levels, we evaluated more than 3.9 million Common Gardens predictions, which encompass the evolutionary history and experimental design to attempt to better fill in this domain of applicability of these genomic offset methods. So with this data set, we'll ask 3 questions for the talk today. There were 6 in the manuscript, but I'll talk about 3 today. The first is, how does the past evolutionary history of the populations affect performance of offset methods? So those are the simulation parameters. Does it matter which genomic markers are used between the all or the adaptive markers? And how well do offset models extrapolate to novel environments outside of the range of environments used in training? So how well can these models predict performance and fitness in increasingly novel climates that we expect to see on the landscape? So for the next few slides, I'm going to present figures very similar to this one. We have performance on the y-axis. Note I've inverted it to create more intuitive relationships with the x-axis. And so for this first question, we looked at those simulation parameters and see how they affected offset methods. And overwhelmingly, it was local adaptation in those populations that drove offset performance. So populations that had high local adaptation had models that performed better on that landscape. And we do see differences among populations. Among those landscapes that I mentioned earlier. And this slide shows each of those genomic offset methods on the x-axis, but what I want you to take from this is that no offset method had similar accuracy across each of these 3 landscapes. And so this stepping stone Klein scenario was the one where almost all of the methods performed best, as we might expect, and that mountain scenario presented some challenges, and that estuary Klein scenario presented further challenges as well. So no method was performed uniformly well across all these landscape scenarios. But local adaptation is the primary influence on performance, and some of the landscapes could reduce performance. And so for this figure, I'm showing a scatter plot that is color-coded by the density of the points that are on top of each other. And so we have upwards of 10,000 points on top of each other, and the black is between 1 and 100. Well, I didn't mean to show that so early, but on the y-axis we have the performance of models using the whole data set of markers, and then performance of data sets that used adaptive markers only. For LFM2, it's pretty symmetric along this 1-to-1 line, indicating that that marker set isn't all that impactful. But for gradient forests And the RDA that's not corrected for population structure, we do see some advantages of identifying adaptive loci. But while they do provide some advantages, they're not universal or by a great margin. So for the last question,
This
we
talk
asked how
is
well
from
do
my
offset
lab,
methods
so I'm
extrapolate to
just in the
novel
interest of
environments outside
time
of the range
going
of environments
to
used in
stop
training.
it so we can move on to the
So
next
let's
speaker.
consider a scenario where we sample populations on the landscape for genetic and climate data.
This question is that they do
given
not
some
extrapolate
forecasts of
well
climate change, we
to
know
new
some of these
novel
populations
environments.
Inhabit novel climate environments.
all right,
that were
Seth,
not
did I
in
s-
the
how do
climate
I
data.
stop
So that
this?
could be in situ related to
I
climate
can't-
change, or this could be for a restoration project trying to transplant
see.
individuals into a climate that's not in the model. And so-
Hi, everyone. Thanks for sticking around for the last session. Can you hear me in the back? Okay, awesome. My name is Brittany Verrico, and I'm a postdoc at UVM, and today I'll be talking about some work that was done to validate genomic offset predictions in the coniferous tree species red spruce. Thank you for the symposium organizers for allowing me to share this work. So climate change is here, and one of the pressing questions is what will be the biological impacts? And one way that we've been assessing this question is through climate forecasting of species distribution models, where these models basically use presence-absence data and climate data to map suitability to some change in the climate in the future. These types of models are a species-wide response, and they basically assume that the same kind of evolutionary rules apply across this entire species distributional range. In other words, they're saying that all populations are the same. But we know that populations tend to vary genetically across the distribution. And one way that this arises is through spatially heterogeneous selection, leading to local adaptation, where phenotypes within a population become more fit and adaptive in their home environment compared to elsewhere. And we can see that it's like really hard to do this and not be able to point, but through the fitness distributions along the environmental gradient, and we see that the fitness peaks happen at different points along that climatic niche. And then with that, the genetic composition can be structured along the same environmental gradient, such that specific genetic variation can then be linked to specific parts of the climatic space. And so as Rebecca kind of talked about, can we marry these kind of two approaches, more ecologically based, say species distribution modeling, with genomic regions? And so as has been previously discussed, there's like a new field or subdiscipline with these forecasting models, and today I'm going to talk about genomic offset models specifically. So as Brandon alluded to, these models aim to forecast the magnitude of local maladaptation expected from the decoupling of the gene-environment association. And these mismatches or decoupling can arise temporally through climate change, so if the population doesn't move but the climate does, or spatially, say through biological invasions or assisted migration. And so I just want to take a second to walk through the concept. So on the x-axis here, we have the climate gradient. I love temperature, so think of it as temperature. And on the y-axis, we have some adaptive genetic component. This could be one loci. Often it's a set of adaptive loci. And the curve here reflects how selection favors different alleles, and the allele frequencies change along this gradient. So let's say we have two populations, A and B. And they exist somewhere along this function because they are inhabiting different parts of the climate gradient. And so where we see that the points are, or the lines are, this is how the population experiences the climate and the set of alleles that this population has to survive there. One key assumption of these methods is that the species is locally adapted. So again, this is just going on the idea that these are the adaptive set of loci. But if that population is transferred to a different site, or say the population doesn't move but the climate does, then we should see a shift in the genetics needed for that population to maintain that GEA environment or GEA association and survive in that location. And so it's not so much the change on the climate gradient that we're interested in terms of genomic offset, but it's the change in the genetics, so that y-axis that is genomic offset, or the displacement of locally adaptive loci between the current and future changes in the environment. But how can we tell if these predictions are any good? So as Brandon mentioned, to validate these, it's a lot of work and takes a lot of time. And so simulation data is great. And then another kind of side to that is the empirical validation with ground truthing in situ in the field. And so here we're looking for the relationship between genomic offsets and observations of performance or fitness-related traits, often done in common gardens or in the tree world provenance trials. And so we're testing for this negative relationship where as your maladaptation increases or your genomic offset increases, you would expect performance traits or your fitness to decline. We can take that one step further and do empirical validation with independent training and testing data sets. So the two main sources of data going into these models are genomic data and trait data. So an independent training data set means you have some sort or you have a set of sources going into your genomics, building your gradient forest, or not necessarily gradient forest, your genomic offset method or model, and your testing data set are your phenotypes. So if your sources are not coming from the same populations, then you have non-independent training and testing data sets. And this really matters because offsets for a single species can change for a multitude of reasons. One, what populations you've sampled, not in terms of, just population sampled, time and duration of the common garden experiment, the location of that common garden and where you sampled in the climate space, and also the life stage history or what kind of window you're looking at in your organism. And so some studies have begun to ground truth genomic offset predictions. But not a lot of studies that I'm aware of have done an empirical validation with independent training and testing data sets. And that's what we sought to do here in the coniferous tree species red spruce. So a big question that I hope to answer is, can we use these data sets to validate predictions? And by leveraging multiple common gardens in this particular study that were started at different time points, how does prediction accuracy vary with different fitness proxies? So red spruce is a temperate tree found across the eastern parts of North America, and the range is highlighted here in red. It is often found in cool montane environments, though it can inhabit bogs as well. On the left-hand side, we have exome genomic data collected from 64 seed sources distributed across the range. And those seed sources, we then got the seeds and planted them in 3 replicated common gardens shown in the white text along the range as well, and then measured phenotypes on 2-year-old seedlings at each of those common gardens. The second data set that we're working with is using genomic data from 12 seed sources. So importantly, they're still stratified across the distributional range, but there's obviously less of them, and so it's more sparse sampling, and the phenotypes were collected on 60-year-old trees grown in 1 common garden at the northern part of the range. So the key kind of things here to remember: blue, 64 seed sources, seedlings; yellow, 12 seed sources, adults. If anyone has any good ways to make this succinct and memorable, love to chat afterwards. So then we use that whole exome capture data set to identify some putative loci in that climate adaptation response. And so after the traditional bioinformatic filtering, we ran a gradient forest model with 11 climate variables that have been shown to characterize the climate space of red spruce. We selected the top 0.2% of loci with the highest climate correlation and used those candidate loci in building our genomic offset model. So we are using adaptive loci, or putatively adapted loci. And with that comes climate data. So the source populations, we are using 30-year averages from 1960 to 1990 to characterize the climate space at the maternal tree involved. And then the climate data for the common gardens varies temporally. If we're working with the seedlings, it's 2 years of climate data, and the trees would be 60 years of climate data. And then for the fully reciprocal offset predictions, so for each genomic data set, which I'm showing here from a strand of DNA, we train a gradient forest model using the climate predictors and generate offsets for common gardens planted with the same members of the same population. So this is the within set, the non-independent sets. And then we do the same thing for members planted or sources from that weren't used in the training data set. And then again, tested for that negative correlation between the fitness traits and genomic offset. So for some results, I just want to orient you to the axes. We have genomic offset. It says genetic, it should be genomic. Offset on the x-axis here. And on the y-axis, we have the trait of interest. So on the left, we have genomic data from the 64 seed sources predicting traits from the 12 seed sources. The first panel is diameter at breast height, so how wide the tree is in circumference, the height of the tree, and population survivorship after 60 years. And what we can see is that the genomic offsets really aren't associated with tree growth, but we do see a moderate negative relationship. They're not significant with survivorship. On the other side, we have the reciprocal comparison, where we have genomic data from the 12 sources predicting traits from the 64 sources. And what we see is that seedling height growth really does reduce. We do see that negative correlation between height growth and genomic offsets in this scenario. And that relationship, while the slopes are different, is consistent and significant regardless of what common garden we use. I just want to put this up here. This is the comparison with the non-independent data sets, so the within population comparisons. The results are very much very similar, so I'm not going to spend too much time talking about that. So can we use independent training and testing data sets to validate predictions? Given my, we're all working in case studies in one way or another a lot of times, given my set of populations, I would say yes. Both training and testing, whether you're independent or non-independent, seem to do pretty well. And this relationship is particularly notable for the seedlings. So it seems that regardless if you have 12 sources distributed across the range or 64 distributed across the range, the amount of genomic data in there doesn't really seem to matter. But I think what does matter is that you capture that climatic gradient. So it can't just be 12 random populations. It has to be, you still have to capture that space in order for your non-independent training and testing data sets to work. But the response may be limited by sample size. This is true in all of science. So in the testing sample size, when we just have 12 sources in the Common Garden, we might be power limited. And we're currently working on down sampling the 64 sources to see if that kind of relationship that's on the left-hand side here is still present whenever we have a lower sample size. And it might be trait specific, which again is often the case in these types of models where we don't really see it with tree growth in the 60-year-old trees, but we do see it with survivorship. And I think I have a few minutes left, so I just want to say, while sample size may be an issue in the validations here, and it might be trait specific, I do think there is a biological realm to this as well, and why we might be seeing what we see in terms of the results. So we do see that genomic offsets better explain a reduction in performance in early life stages of the tree, or of red spruce. And in a way, this makes sense. At the seedling stage, we know in trees that selection is really strong and that those seedlings are to become established, and they really have to fight to make it to the understory and then persist and then get to the canopy. And so small differences in height, whether it's 10 centimeters or even 20, may have an impact and lead to a competitive advantage in those early kind of windows. But why doesn't this relationship persist for later life stages? There could be a few reasons. One is, did we miss this window when selection is strongest? So are we just a little too late with 60 years? Did the sources kind of all catch up to each other? Because at some point, if you're established in the canopy, you just grow, and then what's ultimately going to kill you is some extreme event, or in this case, porcupine wreaking havoc in the Common Garden. But we may have missed some of that phenotypic variation that was present before. And in trees, we know that diameter and height asymptote with age. And so one way to get around this is to look at fitness proxies beyond the seedling stage, but before reproductive maturity, to kind of mind the gap between the two. And to see if there are certain life stages that are particularly vulnerable to maladaptation. And this is extremely important when you're building your models, because while the trees need, long-lived species, trees in particular, need to persist in that environment, they have to establish in that environment as well in order to live long. And so one way that we can do this is estimate and validate genomic offsets with dendrochronology. So we can use the annual tree rings to gain an insight on a yearly basis to see how performance is doing with genomic offsets. When we see, like in this figure here, I'm plotting basal area increment, we see a lot of noise from when the trees were first planted until about 1994. So is that the window? Is that, pre-reproductive maturity, the window in which we should be making genomic offset predictions in trees? Up for discussion and would love to chat about it. I think I'm out of time, but I would like to thank my collaborators, which none of this work would be possible without: Steve Keller, Matt Fitzpatrick, Thibaut Kaplunk, Susanna Lockmuth, funding, and a slew of other people. And thank you for coming.
Well, nothing's done. The bell hasn't done yet, but I'm still gonna just get started. Sorry, guys, I lost my voice 2 days ago, so can you hear me in the back okay? All good? Wonderful. Okay, so we've been talking a lot today about responses in mean annual temperatures, the main thing that the public looks at with climate change over time. But what I want to put out there is that the extreme events that we're experiencing from climate change are equally or even more important than that change in minimum in mean temperatures over time. And we have things like heat waves that are increasing both in terms of frequency and in terms of the anomaly that they propose. So they're getting more severe as well, too. And the responses that organisms have to those different changes are unpredictable in some sense as well, too. So this is the species I study. You can see the yellow all on that little hill over there. That's the monkey flowers I'm studying there. That's a typical year. This is IM, one of the most diverse plant populations that we know of. This is 2 years later on the exact same day. Notice that, well, a couple days later, we notice there is no yellow right there. And that's because there was a heat wave less than 10 days after the start of the growing season up there. It's a 6-week growing season, and we only had 8 plants that year that produced seed when there's normally about 10,000 in that population that produce seed. All right, so what I'm going to talk to you today about is the silver linings hypothesis, the thought that when you expose populations to these severe selection events, that the selection that's actually imposed in those populations is going to make these populations better when they experience future events. So you can put that this way. If you see an extreme year, for instance, in our monkey flower populations, we're going to see a decline in fitness in that individual year. But those plants that are less are the strong ones. They're the ones that can resist, that can tolerate, that can escape that particular stress. But can that selection in years later actually result in higher fitness when they experience a similar selection pressure again? That would be a silver lining to that original heat wave. Now, not surprisingly, because this requires quite a bit of data and baseline data on these different populations, we have very few examples or limited support in natural populations, just mainly resurrection experiments and things like that within plants. And I'm going to say there might be a different reason, too. I'm going to say that this selection that's imposed by these heat waves and these strong events may not always be adaptive afterwards. Sometimes it may be in the wrong direction, and it may result in an evolutionary trap. So for instance, for that extreme heat wave, if we see, come on. Mouse time. All right, there we go. Let's say we see directional selection on flowering time in that extreme heat wave year, in that 2019 year. But it's for later flowering time because that selection event was at the beginning of the growing season. Well, we know that these populations only have 6-week growing season in the first place. That later flowering time is actually going to be a large detriment in later years, right? So, evolutionary trap. There will also be transgenerational maternal effects that could take place here too. For instance, that heat wave could cause lower seed sizes that affects germination ability. Slower germination or less germination would again be an evolutionary trap in future generations. So I'm asking today, in a series of different years where we observe fitness changes in evolutionary populations, do these extreme events impact populations? Do they cause strong selection? Are there maternal effects associated with these extreme events? And are they adaptive in the following years afterwards? And the species we're using is Mimulus guttatus, or the common monkey flower. We study this largely because it has a wide geographic range and it's well adapted. It's a good model for studying adaptation. It has tons of genomic resources, a great genome that's annotated, and it's a really nice model system. What you need to know about it for this particular experiment is that the inland annual monkey flowers that I study, their population dynamics are controlled by water. Their growing seasons start at snowmelt or when spring rains come, and they end in terminal droughts at the end of the year. So throughout the entire the range, there is severe variation in the timing and duration of the growing season, such that some of the populations, the one that I study, are actually threatened in some ways. So this actually makes kind of a nice system for climate change research as well. In the high elevation Oregon populations I study, where there's a 6 to 8 week growing season, we see an adaptation leg that's formed. So in this different populations at this high elevation site, we see, for instance, that high elevation California populations that are hotter and drier with earlier growing seasons do better than the native populations in these sites. This is consistent throughout multiple different years with many different populations that we've used. So when I started my professorship, what I wanted to do is I wanted to start tracking these experiments, see if they were actually threatened. So I started going out every year in 2018 and collecting from 12 different populations in just a 25 kilometer square grid out here. Every year, we go and have transect lines. We collect fitness on those transect lines, 50 to 100 plants. We went down to 50 because they were more threatened than what we thought they were. We started planting them out in common gardens, and we take genetic data from this. We'd be able to get seed masses, ability to germinate, germination speed, and then we measure a bunch of phenotypes in the offspring to look at how that changes over time as well. Okay, so let's get to some data. Right, so these are the 5 years in terms of weather conditions that you're seeing. You should be able to see a few things. Look at these little arrows in 2019. The solid line is the historical average for 1980 to 2010. So not really history, right? And each of these lines are 1 standard deviation or 2 standard deviations above that historical average. So what you're seeing in this white region represents the growing season in June and July for Guttatus. That's that 2019 heat wave that I talked about earlier. In 2021, we had the hottest day on record in Oregon coming at the end of June. This was this heat dome event that any of you in the Pacific Northwest certainly would have experienced. 120 degrees Fahrenheit in Seattle. Absolutely crazy. If you look at mortality in our populations through those time periods, in 2019, that early season heat wave, we had over 50% mortality on average across the populations we surveyed. And 23 of those 24 populations, 23 of 24 grids that we surveyed went down in number during that heat wave when they're normally increasing in frequency. In 2021, this is before the heat wave. This is in the middle, right after that big peak. You can see there's a lot less yellow there. But by July 10, which is about a week earlier than they normally die off, everything was absolutely gone from that population. So notice that the timing and intensity of these heat waves differed a little bit, and so our expectations would too, but they both caused a lot of mortality. If we look at fecundity data, you can see that there's clear decreases in 2019 and 2021, but it's really heterogeneous across populations. Some populations didn't do quite that bad in those different years. We had one population that went completely extirpated. There was complete mortality in 2019. The seed bank caused it to blink back on in 2021, which is kind of interesting here as well too. And you can notice that the surviving individuals still produce a lot of seeds as well too. There is sweepstakes germination here. Not a lot of those seeds actually survive and reproduce later on. Okay? So if we're looking at the consequences of those heat waves for mortality and just survival, what we see is both 2019 and 21 had strong impacts on mortality and fecundity, but they affected populations in slightly different ways in those 2 different years. If we look at selection, so we took all of our populations, we planted a refresher generation, we selfed all the seeds in the refresher generation and grew a second generation common garden to get rid of some of those maternal effects, and then we measured a bunch of different phenotypes in those common gardens. Several of the people that did this, Donna and Andrea, are up here. It was a large effort for the entire lab for this one. And I'm just going to talk about flowering time right now. So if I show flowering times, this grass looks messy. But remember, this is flowering time shifting from 30 days after germination to 60 days after germination in a species that has a 6 to 8 week growing period. And I'm going to make this a little bit clearer too by looking at 2019 and the different populations that have the greatest effects. So in 2019, what you can see in 2019 is there's some populations that had selection for much later flowering. We're talking over 10 days later in flowering time from one generation of selection in these gigantic populations. Other populations had selection for earlier flowering. Now, if you're thinking about this with some populations with earlier and some populations with later, this cannot both be adaptive in those same populations over time. And in fact, this is going to be detrimental to one of those two populations. In 2021, it was more uniform. Almost every population was selected for earlier flowering because the heat wave came a little bit later in the year. This is what you would think of for things that have silver linings. These are not all going to have silver linings, and that's the moral of my talk right now. Okay, let's look at maternal effects. So we didn't even think about this really before we started seeing this pattern. These seeds are tiny. You don't think of them being very big in the first place anyway. But in that 2021 heat wave, our seeds declined by 34% in terms of their average seed mass, which is incredibly substantial. And that, we don't know if it was because of decreased maternal provisioning, because they got that stress, or if it was because for selection for smaller seeds. We don't know. But that affected the ability to germinate. We saw massive declines in the ability to germinate at all. And we see some populations, that HDM population that went extinct, that is just in a meltdown cycle, essentially, right now. Interestingly, germination speed didn't decline in 2021 after the heat wave, but it did decline in 2022. This could be a cohort effect. This could be a lag effect. We don't actually know. But the germination speed is 55% slower in 2022 than it was earlier in the experiments. So if you're looking at this, we see declines in seed mass in 2021, but not 19. We see declines in germination ability in 2021, but not 19. And then we see weird things happening with germination speed. So there are both maternal effects and evolution occurring within these populations at the same time. So let's connect it. Let's try to connect and see how those two things relate to all these climate variables and to each other, and to see if it's an additive effect on their ability to be threatened here. So we made piecewise structural equation models. We used generalized additive models to account for nonlinear effects. We had random effects or smooths for populations, and we had correlated errors between seed and germination traits because we don't know directionality on these. Mark Janung was the primary driver of this modeler. He's an ecological modeler. And we built these different models. The full model includes climate effects on both seed traits and germination traits, as well as seed on germination. And then we take away one of those two things and see if those models fit better or not. So we take away the effect of climate on germination speed, or seed weight on germination speed, or climate on seed weight. Do those models fit better? Turns out they do not. The full model is the best model here. And once we refine that model as well, too, we see something like this. Now, this is busy. This isn't ecology, so you aren't expected to actually know what all this actually means. But if you look at all those climate variables and look at the number of seeds and germination speed, what you can see is the same colors are going from those different climate variables to the germination speed and the number of seed traits over there. And I'll break down a little bit more in here. If you just take July average temperature, for instance, when it gets hotter, germination speeds get slower. When it gets hotter in July, number of seeds and fecundity declines. When it gets hotter, these populations have less ability to germinate, more seed dormancy, essentially. So despite the fact on average that growing seasons are getting earlier by over 2 weeks in the system, we're seeing these effects that are going to be problematic for the system. Thus, that selection seems like it's an evolutionary trap rather than a silver lining in the system. Okay, is this actually adaptive in the next generations? You have to do experiments in the field and manipulative experiments in the field to actually figure this out. Again, large lab-wide projects over here. We got 4,000 plants in the ground right now from all those years and times that we're surveying in dry and wet environments to mimic those different heat waves and see if it's actually an advantage or not to have those different heat waves. So my conclusions are, the heat waves result in increased mortality and losses of fecundity and even complete mortality in a single population. It's heterogeneous across this metapopulation, so the entire metapopulation is not going to go extinct here for sure. And the timing and severity of those heat waves make large impacts on their actual effects. The later heat dome had different effects than the earlier heat wave. In general, selection and maternal effects may occur in opposite directions expected for changing success in climate change. So evolutionary traps may be as likely as silver lining, and we need to take this into consideration when we're doing this modeling. Further, if we're going to be developing models and thinking about genomic offsets, as almost all the talks have been here, we need to go out in the field, as Brittany was doing in the last section, and actually justify these things in the field and validate these things. We have to consider fluctuating climates, and we have to consider extreme events, which may have disproportionate effects on these different populations. With that, thank you to the people that go out with me in the mountains, that do bioinformatics with me, and sit in the lab and do all the molecular genetic work, because they're the people that really deserve this. Thanks to NSF, and I got a postdoc available. I'll take any questions just for a second.
Thank you, everybody, for coming. My name's Daniel Anstett, and I'm going to be talking about evolutionary rescue and trying to potentially predict it. I'm going to be leaning over because I don't have slides in front of me. They're only up there. In California, there's an extreme drought that happened in the 2010s. And you can see here on the left, we have a lot of water that's available in the reservoir. On the right, we basically have no water available and next to no water available. And as you can imagine, that can impose a lot of stress on plants that are already adapted to these riparian systems. And so this is illustrated here in terms of drought monitor at every 2-year intervals here, where we have extreme and exceptional drought that's becoming unfortunately less extreme and exceptional. But in fact, it's this four-year window from 2012 into 2015 and then tapering off in 2016, that it wasn't any one year, but the combination of years in total that gave a drought impact that was the most predicted in over 10,000 years of potential records. And so a question is, can our population is going to go extinct? Are they going to be able to plastically respond? And ultimately, are they going to be able to rapidly adapt to these extreme conditions, given this extreme selection pressure? If that happens, we have here a cartoon where potential drought-adapted allele or trait in red kind of increases in frequency over repeated bouts of selection that are happening each of these years, and then potentially leading to population recovery and evolutionary rescue. Now, the questions I specifically want to ask are, does evolutionary rescue, here, I'll stand over here so I can actually read my slides. Does evolutionary rescue increase in spawn occur during climate change? And then the second one is, can we ultimately use some of this genetic diversity to try to predict when evolutionary rescue might be happening? Can everyone still hear me? Yeah. Cool. So for every evolutionary rescue to occur, we need 3 main ingredients. First of all, we need to have the population declining. And in this case, we're thinking about climate change, so it's declining in response to climate change. Next, we have that evolutionary, during this time period, we have population size here, and then we have allele frequency. So we have potentially SNPs that are associated with climate adaptation or traits increasing in frequency during that time period. And then finally, to kind of get the third potential bit of evidence, is that we ultimately have this rapid evolution predicting predicting the population recovery. And so I'm doing this within the system Mimulus cardinalis, or scarlet monkeyflower. We already had one Mimulus talk already, so I'm glad to be in good company here. This plant really it grows along seeps in areas that have a small amounts of flowing water into medium amounts, so really does require this flowing water in order to complete its life cycle. If you don't water it, it tends to die in the greenhouse and certainly in the field as well. And so it's a really good indicator of the impacts of drought. So in terms of the actual conditions that are happening, there is a wide variety of different types of habitats, ranging from wetter habitats in the northern part of the range on the far upper left, all the way to more drier habitats around LA and Southern California. So there's quite a bit of a latitudinal gradient already happening. And because of this, there's potential to really understand adaptation and impacts. And so this work has really been in collaboration with Amy Ingert and Seema Sheath, who's here in the audience, where they started really by Amy going and sampling this demographic data year after year, range-wide, and really truly building this remarkable data set. where the demographic fates of individual plants are tracked over time. So we go back year after year, and we end up with this information on population growth rate, the lambda that's estimated from a variety of different life history stages within the plant. So ultimately, the plant's ability to grow in future years, where not all individuals are the same, where younger individuals have more potential to contribute more to the population. In terms of what happened during the drought itself, we had, if we look here, we have in 2010, the pre-drought period, then we have a period of intense drought around 2014, and then the recovery. And sort of tracking this in this northern population, we have the kind of stereotypical decline and then recovery of evolutionary rescue. But you'll notice there's a little dotted line, and that's kind of the rate of replacement. For the most part, we're not really dipping down as much in terms of the rate of replacement. These populations are under less risk of becoming extirpated. And that compares with other parts of the range where we're in fact getting this dip, certainly in the center, and in one population, the south even went locally extinct for several years before it was later recolonized. And so we're certainly seeing a lot of variability that's associated with this drought period itself. This directly did correlate to variables such as amount of precipitation that occurs during the winter months that might be in fact critical to the water flow and the formation of snowpacks that would then feed these streams and seeps during the rest of the year. Next, we need to figure out if there's been evidence of rapid evolution. And so in order to do that, we have, I carried out landscape genomics on many sites throughout the range at a baseline level before the start of the drought. So this was 2010. We happen to have, fortuitously, have all these collections that were done from 2008 to 2011. And from there, I carried out whole genome sequencing on just around 350 individuals in total, and then ultimately put it through the pipeline and eventually got to individual SNPs and variant calling. And then, because we have this latitudinal gradient, we can do a space for time substitution, or the idea that ultimately we have a cline that we're looking at, and ultimately we have these climate relationships over space that can be approximated over time. And there's many ways to do this. One possible way is to associate all these SNPs over time and do a genotype environment association to ultimately say here on the left we have a SNP that's associated with greater precipitation, or on the right another one, kind of the inverse, that would be associated with less precipitation, naturally, historically, before the onset of the drought. We take this information and then we do this across many environmental variables and ultimately come up with two different approaches, a windows-based approach, a SNP-based approach, BayPass, and WIS. I'm happy to go into more details if there's interest. But effectively, we find 600 SNPs where we have the best possible evidence for association with variables such as high temperature, low precipitation, water availability, or moisture deficit. Once this is done, this is half of the genomic data. The other half, again, whole genome sequencing on temporal genomics. This time we have 400 genomes that I was able to sequence across 12 populations over 6 years. You can imagine sample size dwindles, but really the power is having many different populations where we have potential to see repeated instances of rapid evolution. And so then this looks something like this. For each of these SNPs, they're going to be changing over this year, and we can have a variety of different slopes, where a positive slope would indicate approaching towards 1, which is the climate change associated state. I've parameterized all of the different climate variables to be that way. And a decline would be kind of away from the predicted state, at least according to genotype environment associations. And so this is done. This looks a little bit like abstract art. We have 600 different SNPs here. But if you can squint, you can kind of see on the left here, the southern population, we have more of them that are increasing compared to this northern population, where there's some but not quite as many, and there's more declines. But it is messy. This is messy data. I mean, it's ultimately associations. And so we can take this and present it in a form that's more easily understandable to humans by taking the slopes and binning them. And that comes up with something like this, where we have, again, these are just 2 example populations that are kind of, which represent some of what we're seeing in the north versus the south, where the south is seeing potentially more drastic effects and impacts, And then we're seeing potentially some stronger amounts of selection that are occurring, or a longer tail of positive selection towards the climate change associated state compared to this northern population that we have more data closer to the mean. I can be somewhat confident that at least for some of these populations, this is distinct from drift itself, because you can imagine populations are crashing and we could be having drift carrying many of these genes forward, but these alleles forward. But I did some permutation study, which I won't have time to get into today, that ultimately shows that the overall patterns that we're seeing in terms of mean and median selection was outside what you'd expect just by drift alone. Okay, so now we can say that we've put a little check mark now next to rapid evolution occurring. The final step is then ultimately getting more towards really being able to predict the population and demographic impacts. And so in order to do this, I'm able to take the demographic information from the recovery period. So in here, the 2015 into 2017, 2018. And when I do this, I'm able to take the mean of the lambda across those years of the growth rate and put it on one axis, and then take the median strength of selection, so that's the mean there, it's median, put a strength of selection on the other axis. And ultimately, if we have this strength of selection kind of showing that ultimately a lot of adaptation and rapid evolution occurred, if we have more of that, we'd expect that the populations are doing better if there's a causal relationship. And so I took this for the populations where we had the best possible data, and that ultimately led to a pattern like this that's quite convincing, where we have greater median strength of selection, ultimately predicting greater increased populations. There are potentially some influential points, so we can remove those, and the pattern still holds regardless. R squared is now slightly higher. So we can say that we've done kind of done all three, and ultimately shown that evolutionary rescue is occurring in this system across this latitudinal gradient, and that we have some amount of predictability here. However, this is just part of the story, because ultimately many of these systems won't have that kind of data, that kind of resolution, so we might have to predict just with the landscape genomics before a severe event. In which case we can ask if ultimately we can use genetic diversity and potentially the withstanding genetic diversity before the selection event happens to ultimately predict. Which, when we do that, we can do that for the two different SNP sets. We can do that for the SNPs that we could calculate nucleotide diversity, or pi, for the SNPs that were climate associated, or we could calculate it for all the SNPs overall. And when we do that, we'd expect that perhaps the climate-associated SNPs would be better predictive. And that's exactly what I saw, quite a remarkable amount of predictive ability ultimately within this setup. There was one influential point that we ultimately was that we had specific reasons for why the sampling had occurred. I'm happy to go into that, but for like the vast majority of the data, we had kind of this high, tight correlation. That's very different from the nucleotide diversity genome-wide, where we ultimately have not that much predictive power at all. And so ultimately, yes, we're able to show that potentially we have diversity predicting, which more broadly, given the broad concordance of these two patterns, we can say that perhaps we're able to move towards predicting evolutionary rescue using genetic diversity data. Now, a brief plug. Ultimately, we have to understand what's happening in the distant future, and so it's going to be a challenge to really try to bring all these results together to try to predict more distant events, and that's something I will be doing at my new position in Cornell, where I'm starting a lab this fall, and then potentially I'm recruiting students and postdocs, so come talk to me if you're interested. Thank you.
So my name is Donna Hinrichs. I'm a master's candidate at the University of Louisiana at Lafayette, and I'm very excited to share my research that is assessing the consequences of assisted gene flow in threatened populations in order to expand our conservation toolbox in order to best preserve biodiversity in the face of climate change. Now, we're not strangers to the fact that climate change is negatively impacting biodiversity. We're seeing more species than initially predicted going extinct due to a variety of reasons. And the reasons that I like to focus on are the increased severity and frequency of extreme selective events, and simply the introduction of novel climate conditions in an environment at an extremely alarming pace. Now, in the face of climate change, in order to escape extinction or local extirpation, organisms can move, acclimate, or adapt. However, not all organisms are mobile, and acclimation is only a short-term solution to long-term changes in environment. So I'm focused on how organisms can adapt in order to escape the detrimental effects of climate change. But with the current rapid pace of climate change and increased extinction risks, are we able to help speed up the process of adaptation for organisms and populations that have no other option? And we may be able to do that through utilizing assisted gene flow, which I might intermittently abbreviate as AGF. We can move individuals within their range in order to increase genetic diversity and hopefully facilitate adaptation to local conditions. For example, a population at risk in California could potentially benefit from introducing genetic diversity from a population within its range with a historically hotter and drier climate. For example, in this hypothetical scenario, New Mexico. Now, facilitating adaptation through assisted gene flow can allow populations to better tolerate changing conditions. During climate extremes, the introduced genetic diversity could produce fitness increases by being better adapted to those conditions, or populations where assisted gene flow was conducted may take a less severe hit than the other populations, so to speak. Now, with that being said, we also need to look at the variation in fitness after conducting assisted gene flow. We could see increased variation in fitness due to an increase in genetic diversity. Individuals with this new adaptive variation may have significantly higher changes in fitness in changing climates, while individuals without that variation may suffer even greater, increasing that overall range in fitness within a population. Now, while assisted gene flow has the potential to be a critical conservation tactic, there are several risks associated with translocating individuals that has produced some controversy around the topic, including deleterious combinations of introduced and native alleles, otherwise known as outbreeding depression, and introduced genetic diversity having the potential to swamp out that native diversity. Now, despite the controversy surrounding assisted gene flow, there are no experiments that uncover the consequences in threatened natural populations, and that's what my research seeks to understand. So the questions I sought to answer in this portion of my master's thesis are, 1, does assisted gene flow affect mean fitness and variation in fitness? Does growth stage used for introduction alter the fitness effects after assisted gene flow? Are there differential responses between years following assisted gene flow? And lastly, did non-native alleles successfully introgress into our target populations? And can we attribute any differences in fitness that we do see to novel genetic diversity in the target populations? Oh, look, another Mimulus talk. So to answer these questions, we're using populations of Mimulus guttatus, or the common yellow monkey flower, which has a wide geographic range that possesses an incredible amount of phenotypic and genetic diversity across the western United States, has well-developed molecular resources and annotated reference genomes in order to streamline genetic data collection and analyses, and a long history of ecological experiments, as well as rapid generation times to make those experiments just a little bit more manageable. Now, while Mimulus guttatus as a species is not threatened, there are populations and areas throughout its range that are experiencing increased risks of extirpation and even some observed extirpation events. Now, my advisor, Nick Kooyers, kind of set the stage for my research in identifying that the contemporary climate conditions in Oregon populations, such as growth season start dates, are now closer to average historic conditions in California populations than the historic Oregon conditions. Now, taking this into account, we see that in low and high elevation Oregon sites, elevation match populations from California actually have higher fitness than their native Oregon counterparts. This adaptation lag demonstrated in Oregon populations, combined with the discovery of potential source populations in California with higher fitness in our target climate, made these populations great candidates for conducting assisted gene flow and evaluating the consequences on a deeper level. So that's exactly what we did. We conducted assisted gene flow at the beginning of the 2021 growing season. We used 2 populations near the Sierra Nevada in California as our source populations, one at a high elevation, which is labeled Shell, one at a low elevation, which is labeled Bell, and we crossed those to produce F1 source individuals, and those were the individuals used for translocation. For the sake of explaining, I'm going to zoom in on the purple group of populations there at the bottom, and this is one of our 4 experimental blocks, and we repeated the process that I'm about to explain in each block. In our control populations, we dispersed 200 seeds native to that population, so in this block, that's our EVS. In our seeds population, we distributed 200 seeds of our F1 crosses, and that is our MTD population in this block. And in our seedlings population, we germinated seedlings of those F1 crosses in greenhouse conditions, and then planted approximately 20 of those seedlings in this block, BMP. And then we repeated that dispersal in each of our 4 experimental blocks, which are color-coded here. So here's sort of a timeline overview of our data collection over the past 6 years. So at the end of each growing season from 2018 to 2023, we collected and counted flowers and seeds from 50 maternal field lines per population per year, assessing fitness 3 years before and 3 years after conducting assisted gene flow. 30 of those field lines were grown in refresher generations in greenhouse conditions, and DNA was extracted from approximately 24 of those lines for whole genome sequencing. Now, with the fitness data collected from the field, we found that in the control populations, which are demonstrated on the far left, we see virtually no change in flower production before and after assisted gene flow. However, conducting assisted gene flow using seeds, which is shown on the right of this graph, resulted in a 19.4% increase in flower production when compared to the number of flowers produced pre-assisted gene flow. And while we did see a slight increase in flower production in the populations where we conducted AGF with seedlings, it was nowhere near as stark of an increase as in our seeds populations. Now, our second prediction regarding assisted gene flow considered the standard deviation in fitness. And while we don't see a significant AGF by treatment interaction, we do see elevated levels of variation in flower production in both of our experimental treatments compared to our control. On the other hand, we don't really see any statistical signal for an increase in seed production here, but we did see some seed production in our target populations begin to trend upwards ever so slightly. And we see that the same trend was presented when looking at the standard deviation in fitness production. So while we see some increases in flowers and seeds, we noticed a lot of heterogeneity, so we wanted to break this down to see if there were differential responses between our site groups and between years following assisted gene flow. So to do that, we utilize this ratio, this very messy ratio, to qualitatively compare the fitness of experimental populations to their corresponding control population in a given year. So to break this down, the numerator of this ratio is the fitness of an experimental population in the year of interest divided by the mean fitness across all 6 experimental years, and we subtracted from that the mean fitness of the experimental population before assisted gene flow was conducted divided by that same overall mean across all 6 years for that population. And then that difference was divided by the same metric for its corresponding control population in its experimental block. In short, a value greater than 1 indicates that the experimental population had a greater increase in fitness than its corresponding control, while a value less than 1 indicates that the experimental population didn't perform up to the standards set by its control. And with this data, we found that some site groups show increases in flower production immediately following assisted gene flow in both experimental populations compared to their control, and those increases are expanded upon in the following growing season. However, the same populations don't show the same trend when we look at seed production. In this case, only our seedlings population, which is OWC shown in blue, show an increase in seed production relative to its control, and only in 2 of the 3 growing seasons following assisted gene flow. And in some years, neither population produces enough seeds to match the standards set by their control population. This suggests that there's a lot of heterogeneity between years and between the fitness variables that we're measuring, which further suggests that we need to continue assessing yearly changes in fitness and potentially assess the climate conditions that are underlying these differences between years. Now, not only was there heterogeneity among our measures of fitness, but also our different site groups responded to assisted gene flow in different ways. For example, we see that assisted gene flow didn't increase flower production in both MTD and BMP in either year when compared to their control population. But we see that our seeds population had a rapid increase in seed production, and our seedlings population followed closely behind, resulting in increased fitness relative to their control in both populations for the 2 subsequent years. Now, why might we see these differential responses? Well, the first two populations that I showed you, RRM and OWC, were our lowest elevation population block, while these populations that I just showed you, MTD and BMP, are some of our highest elevation populations. And these specifically are separated geographically from the other 9 in this experiment. If you can think back to the map, these two are some of the populations that are lowest geographically. And we can also attribute this to some stochasticity between our different site groups. Now, other site groups exhibited intense heterogeneity in fitness when compared to their control populations. And there are a variety of reasons behind this. For example, HDM, which is shown in pink here, was extirpated for 2 growing seasons before conducting assisted gene flow and had relatively very few individuals to interbreed with the California seeds that we dispersed when it reappeared in 2021. After seeing these changes in fitness following assisted gene flow, we wanted to confirm that these changes are a result of successful integration of the California alleles into our target populations. And to do that, we utilized whole genome sequencing of 872 different lines to generate a data set of alleles private to California. And then we tracked those private alleles through our target populations following assisted gene flow. Now, I'm going to preface that this data is very recent and very preliminary, but it's still pretty interesting, so bear with me. We found approximately 208,000 SNPs that were private to California at an allele frequency above 0.3, with an average of 1,953 base pairs between California private SNPs. And looking at our experimental populations post-assisted gene flow, we do see some evidence for introgression of these private alleles in our target populations, but we also see that what appears to be some introgression into our control populations, but that may indicate some error in the way that we initially called our private alleles. However, the alleles that did successfully introgress into our target populations show an increase in frequency from 2021 to 2022, suggesting that our assisted gene flow experiment successfully introduced some genetic variation into the background here. But there's still much to come from this part of my research, so stay tuned for some future results. Now, in conclusion, we found that assisted gene flow did lead to increases in flower production as well as the variation in flower production. And while it didn't necessarily affect seed production statistically, we do see trends in the right direction. Conducting assisted gene flow with seeds led to greater increases in fitness than seedlings, and responses to assisted gene flow differed significantly between years as well as between site groups. Lastly, we do see some introgression of California private alleles into our target populations at low frequencies. Now, as a result of this research, what have we learned about assisted gene flow in natural populations thus far? Well, while statistically significant, our rapid increases in fitness are still very slight, which might suggest that introducing greater proportions of individuals and utilizing multiple introductions may be beneficial for evolutionary rescue. Environmental heterogeneity can produce a wide variety of variability in site-specific responses, both in the population's initial responses to introduction as well as the temporal heterogeneity among growing seasons, which can produce differential effects. And we need to continue to think carefully about the introgression of non-native alleles into our control populations. Overall, there's still much to learn about the long-term success of assisted gene flow. Will the results be comparable to experimental evolution in the lab? And are our consequences context dependent to the species or even the population level? Or can what we learn be applied on a broader scale? And with that, I'd like to thank all the people and organizations that made this research possible. Thank you to the organizers of the symposium, and I will take any questions if I have time.
All right, hello everybody. I'm Emily Harmon. I just finished up my PhD at UNC Chapel Hill, and today I'll be telling you about some of the work I did there evaluating the role of plasticity in promoting the persistence of populations in new environments. So, of course, when environments change more rapidly than populations are able to adapt to those changes, they risk going extinct. But many times when the environments change, we see that individuals within that population also change their traits. And they do this through phenotypic plasticity. And if these adaptive changes are if these changes are in an adaptive direction for that new environment, then we would expect those individuals to survive and their lineages to persist. So plasticity can help populations avoid extinction. This idea has been around for a long time. You may know it as a component of various hypotheses like the Baldwin effect, or more recently, the buying time hypothesis. And we are accumulating evidence from nature that is consistent with this idea. So for instance, Behaviorally plastic birds, so those that are better at finding new ways to acquire resources or use tools in new environments, are also less likely to be endangered. But overall, we are lacking direct evidence of relationship between a pre-existing plastic trait that allows for persistence in a new environment. And this is probably because in many cases it's difficult to actually manipulate plasticity. But today I'm going to present you an alternate approach that we hope can show if there's an overwhelming pattern through which we see pre-existing plasticity allowing for persistence in new environments. So to do this, we first want to obtain a lineage that has some sort of pre-existing plasticity that is relevant for fitness, expose these lineages to new environments, and track them for multiple generations to see if plasticity is adaptive in that environment and allows for persistence of the lineage in that environment. By doing this, we can see, first of all, do we have plastic responses variably produced in these new environments, and does variation in plasticity predict variation in persistence? So I'm going to introduce you to the study system that I've developed for this work. It's not mimulus, but I promise it's still cool. This is a rotifer. This is a freshwater zooplankton. If you know anything about rotifers, you probably don't know that some of them are predators, but this one is. It's fairly large, so it likes to eat other rotifers. And it's great for this sort of work because they reproduce really quickly. They double in about 20 hours, and they can reproduce asexually, so we can maintain clonal lineages where the trait changes that we're seeing are almost certainly through plasticity. And the plasticity that we're tracking in Isplankna is really interesting as well. So the first individual that hatches out to start a clonal lineage is what we call an alpha morph. So you can see one here pictured from the front and the side. And this alpha morph is a female, and as a predator, when she eats herbivorous prey, she accumulates vitamin E in her body. We actually have a transgenerational form of plasticity here where the vitamin E that an individual is exposed to in utero determines its phenotype. And at a threshold level of vitamin E, we get development into the beta morph. This beta morph is larger, so it eats larger prey, and it also has these body wall outgrowths that help protect it against cannibalism. So we have this morphological plasticity and two morphs that vary in their resource use, but we also have reproductive plasticity. So I mentioned that there's asexual reproduction in this system, so both alpha morphs on the top and beta morphs on the bottom can reproduce asexually, so they produce diploid eggs that without fertilization become more females. But some beta morphs instead reproduce sexually, so they produce haploid eggs that if unfertilized become males, and if fertilized grow into what becomes the starting female of a new genotype. And these sexually produced embryos are in a cyst and have an obligate period of diapause, and they can withstand environmental conditions like desiccation. So we have this really interesting system with different morphs that vary in size, shape, resource use, life history. To use it to see if plasticity predicts persistence in new environments, we just wanted to make sure that these traits were relevant for fitness. So to do this, we tracked 72 individuals for life, representing 12 different clonal lineages, in just our control laboratory environment where we feed them small prey paramecia. So we first looked at survival of our different morph types, and we see in the yellow circles there that the alpha morphs tend to live longer than either of our types of beta morphs. If we look at reproduction throughout life, we see a couple things. First of all, they reproduce throughout their lifetime, so the longer they live, the more offspring they have. So the alpha morphs tend to have more offspring in these conditions. We also see that the sexually reproducing form doesn't have nearly as many offspring as our asexual forms. But keep in mind that the sexually produced offspring are those diapausing embryos that can withstand environmental stressful conditions. So for our purposes, this tells us that the morph that an individual expresses in a new environment is going to be relevant for its fitness and the persistence of its lineage. So the next step then is we wanted to expose these lineages to some new environments and see what will happen. So we had a lot of environments that I'll tell you sort of some of the different categories that we changed in order to try to select for different morphotypes. So of course we started with our control where they're kept at 20 degrees Celsius and fed paramecia every other day. But some of our environments we swapped out the prey type, we introduced a predator or a competitor, we changed the temperature, we changed the physical environment. And overall, We ended up using 33 different clonal lineages or genotypes here that we exposed in triplicate to 10 to 13 environments over some different temporal rounds. And all of these environments also contain a small amount of vitamin E, so we're expecting to get a variable proportion of alpha and beta morphs expressed in these environments. After about 6 days, which is enough time for, on average, 2 generations to pass, since remember this is a transgenerational type of plasticity, we measured the plastic responses. So our first question was whether there are plastic responses in this new environment. So I'm going to show you a bunch of reaction norms for all of our environments. So here, the trait that we're looking at is the proportion of beta morphs that were produced in an environment. And within each panel, we have control on the left and the new environment on the right. And each clonal population is one of these lines. So you can see what the clones did in the control environment versus their clones in that new environment. And they're color-coded essentially by plasticity here. So the larger the change between the control and the new environment, the more green that line is. So for our purposes, we can see that, yes, we do have variable plastic responses to these new environments overall. And now we can ask whether variation in plasticity in those new environments predicts their persistence in those new environments. So we tracked these populations for a total of 50 days, which is enough time for 20 to 60 overlapping generations. Okay, and each of these environments was in effect a replicate overall, so I'm going to show you just all of our environments pooled together. We measured persistence both as the likelihood that a population persisted for 50 days and also the length of persistence for those that did go extinct. So here we're looking at the probability that a clonal population persisted on the y-axis over time. This is the output of our survival model. And our populations here again are color-coded based on how plastic they were. But this is actually a directional plastic change. So our populations that produced more beta morphs in the new environment than the control environment are in green and did better than our populations that produced fewer beta morphs or more alpha morphs in the new environment than the control environment. So we do see that this pre-existing plastic ability is co-opted in an adaptive way in these new environments. But of course, we're pretty surprised to find that across all the different environments that we used, it really was the beta morph that predicted persistence rather than, per se, the ability to produce one morph or the other. So we can think a little bit about why the beta morphs may have done better. If we compare them to alpha morphs, for instance, we know that they can eat larger prey, they can eat each other, so maybe they have this alternate resource, but that's probably not going to be adaptive across a wide range of environments. So you're probably thinking, well, obviously some beta morphs can reproduce sexually, and sexual reproduction generates genetic recombination, genetic variation that would be adaptive in a new environment. And these sexually produced embryos are those diapausing embryos that can avoid stressful conditions in time or space through dispersal. However, because these are diapausing embryos, they actually did not contribute to our populations. They were still in diapause when we ended the experiment. None of them hatched out. So sexual reproduction can't explain why within our experiment the beta morphs actually persisted better. So I don't have an answer for you, unfortunately, but given what we know about the beta morphs versus the alpha morphs, our hypothesis is that the beta morph is essentially a stress-resistant phenotype that under regular conditions has evolved to have various strategies to deal with changing environments, new resources, and stressful conditions that they're going to want to avoid. So in summary, we found that we do see cases where pre-existing plasticity is differentially expressed across a range of new environments. And in our system in particular, the lineages that express one particular phenotype, the beta morph, both persist longer and are less likely to go extinct. So if we zoom out a bit, we know that plasticity is everywhere. Every organism has some capability to respond to its environment, whether it's through morphology, physiology, or behavior. So it's going to be common that plasticity is expressed in new environments. But where we really need to do more work is figuring out whether that plasticity in a new environment is going to be adaptive or not. And this is going to vary depending on the environment, the type of trait that varies, and whether its adaptive function is conserved from the new environment, or from the old environment into the new environment. And overall, of course, it's been mentioned that plasticity is this kind of short-term response. So what does this tell us about evolutionary responses in new environments? Well, because plasticity allows for population persistence, this can buy time for evolutionary adaptation to the new environment to occur. So the longer a population persists, the more likely it is that we'll get longer-term genetic adaptation to those environments. I'd like to thank you all for coming, and I'm happy to take your questions.